Hello and welcome to our last lesson in the series on life sciences, plant functioning and structure. And in this lesson, I'm going to be revising some of the concepts we dealt with in our earlier lessons and having a look at some examples of questions that might be asked in tests or exams. We are going to be looking at a revision, first of all, of plant tissues, then plant parts, the structure of roots, stems and leaves. Then we're going to be looking at some practice questions. What are the types of questions that can be asked and how do we actually handle those questions? As a revision, there are 4 basic types of plant tissue, and they all have different functions. And we're going to be looking at meristematic tissue, epidermal tissue, ground tissue, and vascular tissue. And remember, you have to be able to identify, if you're given a drawing or a photograph of each of these different types of tissues, you have to be able to identify each of those different types of tissues. The first type of tissue is meristematic tissue, which is your tissue for growth, and it's found in the tips of roots there, in buds in stems, and on the sides of stems so that growth in width can actually occur. In these tissues, the cells divide continually by mitosis to produce new cells. Now, you need to be able to recognize this type of tissue. So if you're given a picture like this, you can tell straight away it is meristematic tissue. Because if you look, the pink things are the nuclei of the cells. And there are lots of pink things very close together, meaning the nuclei are quite large and the amount of cytoplasm is actually quite small. That's a giveaway. Then the next giveaway, and this picture here is an enlarged version of the picture here, but it looks a different color because it's stained with a different stain. And if you have a look, you can see that that cell is busy undergoing a stage of mitosis. So those are the 2 things you look for, stages of mitosis, closely packed cells. Next type of tissue is epidermal tissue, which forms the outer covering of all the parts of the plant. Sometimes it has a cuticle, and that is when its function is protection. If we look at this picture, that's our nice thick cuticle. Sorry about that. That's our nice thick cuticle. There, we're back again. That's our nice thick cuticle on the outside of the epidermis. But some of the cells are also elongated to increase the surface area for absorbing water, for example, your root hair. And on the leaves, some of the cells are stomata with 2 guard cells to control substances moving in and out of the leaf. Then, filling the cells, or filling the organs, we have different ground tissues. And there are 3 different types of ground tissues: parenchyma, thin walls; collenchyma, walls with thickened corners; sclerenchyma, cells with extremely thick walls all the way around. And depending on where they are in the plant, they have different functions. You need to be able to identify them, and those cells there are collenchyma cells because they have thickened corners. These cells here are parenchyma cells because they're large and they've got thin walls, whereas these cells here are sclerenchyma cells because you can quite clearly see all of the extremely thick walls. Then, last type, vascular tissue, xylem, which transports water, and phloem, which transports food. And if you look at pictures, whenever you see anything with thickenings like this, round or spiral, that's got to be xylem. Long vessels joined end to end with thickened walls, and this is phloem. And one way you can recognize phloem is that thing is one of the sieve plates, because the phloem cells are long and thin and they end in sieve plates. Then, in a plant, we have got different organs, roots, stems, and leaves, and each one of those organs has a different role to carry out. And inside each one of those organs, they would have a certain arrangement of tissues depending on the function of that particular part. So what we're going to look at now is the structure of the roots, stems, and leaves. And remember, it's linked to the function that they have. Okay, this is our root, and hopefully this looks familiar to you. One giveaway, of course, is the fact that if it's got those little things sticking out, it has root hairs. That is the cortex of parenchyma cells where food is stored. Then we have these two layers, the endodermis, which we've looked at how it controls substances moving into the vascular cylinder, and then the pericycle. Then, in the center, we have the xylem in the form of a star, and then the patches of phloem. Next, we're looking at a plant stem. It's still round, but looks very, very different. The most noticeable things are these things, which are the vascular bundles. It still has an epidermis, but the cells under the epidermis, it looks quite dark, and that is because those cells have thickened walls because stems grow upwards, so they need a little bit of support. Then, filling the center of the stem, we have great big parenchyma cells for storage, which is the pith. We're now going to have a more close look at a vascular bundle inside a plant stem. This is magnified much more than the last picture, and we can clearly see the epidermis, and if we look just underneath the epidermis in the cortex are cells with extremely thickened corners, and that would be your collenchyma. Then, on the outer part is the bundle cap. That is the phloem. Then that is the layer of meristematic cells called the cambium. And at the bottom, our very, very big cells with thick walls, which are your xylem vessels. Next, we're looking at the leaf, and this is what the leaf looks like. If we look at it in cross-section, you can clearly see the nice cuticle covering the epidermis. In the epidermis, mainly on the lower surface, there are guard cells so that gas can move in and out, and then these patches inside the leaf, those are vascular bundles with xylem and phloem. The rest of the leaf is filled with mesophyll cells, and there are two types of mesophyll cells. Palisade mesophyll, closely packed, elongated with lots of chloroplasts because their main function is photosynthesis, and then the lower layers which are the spongy mesophyll cells, and they've got lots of air spaces because their function is gaseous exchange. After the break, we're going to be looking at some practice questions. How are questions on this particular section asked, and how do you answer them? Now, on this section, we find different types of questions can actually be asked. Most of the questions link the different aspects of plant structure and functioning together. So let's have a look at how questions can be linked. Okay, first of all, there will always be some simple identification. For example, you're shown a picture of cells like this, and you have to say what these are. You also have, so that's pictures of tissues, and it's also pictures of plant organs, roots, stems, or leaves, and that's simple labeling and identification. Then things get a little bit more complicated because now you've got to link the tissues and where they are found in the plant. Then it gets even more complicated because you're now linking tissues and plant organs to functioning. What do these parts do? And then making it even more complicated is you sometimes have to link the parts you have studied to experiments that are done on how the different parts of the plants function. And we've got some examples of each one of these to have a look at. Most exams start with fairly straightforward questions, and this is one of the straightforward questions. For example, you're given a picture and you're asked, what is this picture about? So let's have a closer look. You're asked, what is this? Is it xylem? Is it a stoma? Is it a stone cell? Or is it phloem? So we have a look. How can we tell? This is oval, and it's got two things that look like lips. So obviously, correct answer is B, it is a stoma. Then things get a little bit more complicated because instead of just one thing, you are now given many things and you have to work out what is going on. And the best way to answer something like this is to look at each one, look at the first one and eliminate them. For example, T. Okay, now unfortunately that's not shown too well, but that is T. Okay, so it's pointing to a small thing in the epidermis. So we can tell straight away, okay, it's not A. P, is P the epidermis? Yes, so it could possibly be that. Q, is Q palisade mesophyll? So it could possibly be that. So those are possible. So then we look at the next one. Okay, P is the epidermis. Q is the palisade layer, yes it is, and S is the spongy mesophyll, yes. Okay, and once you've determined that this is the correct one, you actually don't need to go any further. If we looked, we would find it says T is the spongy mesophyll, so that makes it incorrect. And R is spongy mesophyll, nope, because R is pointing to the vascular bundle. Okay, so this takes more thinking than simply identifying. Then you also sometimes find they throw in quite a lot of things together. For example, this one is looking at 4 different types of tissues. So you have to be looking at all of these at the same amount of time and identify them. Okay, starts off, easy questions. Identify tissues A, B, C, and D. And A, it's got big cells and small little cells. That's obviously phloem. B, big cells with air spaces, obviously parenchyma. C, obviously xylem. And D, obviously meristematic cells. It's obviously the meristem. Okay, then it goes further. It says what is the function of tissues A and C. Tissue A, it's phloem, obviously food transport. And tissue C, xylem, water transport. Where would you find tissue D? So this is linking to its position in the plant, so that would obviously be at the tips of roots and stems. And then identify cells X and Y, which is sieve tube cells and companion cells. So even though this looks complicated, what is being asked is really not that complicated. Then, another way questions can be asked is you are given a diagram or a photograph of part of a plant, and you have to identify the types of cells found in it. Is it a root? Is it a stem? And what are the functions of the different parts? So what we have here is, starting off, simple. Which plant organ is shown? Giveaway, of course, this thing here. So it's obviously a root. Layer B would obviously be the epidermis. Then cell A, identified, it's a root hair. And how is it adapted for its function? So this takes you further than simply being able to identify it, is why it is so efficient for its function, and that of course is the fact that it has an increased surface area so it can absorb as much water as possible. Then identify tissue D. Okay, and tissue D here, easy to work out, towards the center of the root, nice thick walls, it's got to be xylem. So here you would have to go back to the information on plant tissues and say how xylem is adapted, for example, has no cross walls, has thickened walls, has no cell contents, things like that. And then the last one is quite easy, identify the process by which water moves. That is obviously osmosis. Next question. Oh, these photographs are actually photographs taken under an electron microscope, and they look quite different to photographs taken under a light microscope. But whatever way the photographs are taken, you need to look for what tells me what type of tissue this is. And of course, for number A, it's a big hole. Okay, so that's obviously xylem because of the thickened. You can see the thickenings. And B is obviously a sieve plate, so that type of tissue is obviously phloem. So this starts off easy, identifying and giving the main function of each, which is fine. Okay, then sometimes questions get a little bit more complicated because they're saying tissue A and B do not have nuclei, but we say tissue A is non-living and tissue B is living. How can it be living if it doesn't have a nucleus? Okay, so for this, you need to understand, because in phloem, look there, what do we have there? We have a companion cell. Okay, so phloem sieve tubes are living. The nucleus that controls them is in the companion cell, and the key is they do have cytoplasm. Whereas Xylem, absolutely hollow. That's why it's described as non-living. And then the last question, calculate the magnification. And this is actually a question from your section on cells, but very often they bring in some questions on cells into the questions on plant tissues. What we've looked at so far is the types of questions that can be asked linking plant tissues and the structure of plants. For example, linking plant tissues to the structure of plants, and we've also looked at some practice questions. However, we will find that sometimes the structure of plants can also be linked to functioning, but that comes in the next part of a lesson. I'm sure your brains are bursting by now, so let's take a break. Welcome back from your break. I hope your brains are fresh, because now things are going to get a little more complicated. Okay, what we've done so far is we've looked at the types of tissue and the structure of the plant parts. And what we're going to be looking at now is we're going to be linking that to how the different parts of the plant actually function. So we're going to start off by revising plant functioning. And almost everything a plant does is based on water. And in previous lessons, we looked at how water goes in through the root, goes all the way up, and it goes out again through the leaves. The different tissues involved are water entering the root hairs here, going through the cortex, into the xylem, being pushed up by root pressure, being kept together by capillarity, and ending up inside the xylem vessels in the root. This shows the tissues involved, how water moves in through the root hair into the xylem, how it's pushed up by root pressure, how it moves up by capillary action, and how the water actually eventually gets into the xylem in the leaf, and it then moves out through the stomata. We're going to have a look briefly at each step. How is water absorbed? In through the root hairs, moves either through the cells, into the xylem, or moves along the cell walls. Same thing happens, also goes into the xylem. Once it's in the xylem, there's osmosis pushing the water up through root pressure. It moves through the xylem, through capillarity, and it ends up in the leaf, and it exits the leaf, and this causes transpiration, pull. What actually happens in the leaf is the water comes out of the xylem, goes into the cells, into the air spaces, and eventually moves out through the leaf in transpiration. Then, when we did this section, we also looked at how plants can be adapted so that they don't lose so much water through transpiration, and there are different ways that they can do this. They retain water vapor, and an example there is a sunken stomata. They also have hairs. They also prevent water loss by having a thick cuticle. They have as few stomata as possible by the leaves being small, or they have their stomata mainly on the leaf lower surface where it doesn't get quite so hot. Then we also find that the environmental factors affect how quickly transpiration occurs, and in our previous lessons we had a look at some of these and how they affect transpiration. And the factors that affect the transpiration are temperature, light intensity, wind, and humidity. And we did look at some experiments on these. The last aspect we're looking at about how substances are transported around the plant is the translocation of food. We know that water goes up to the leaves use the water to make food. Well, what happens to the food then? So here we have a leaf cell which has made food during photosynthesis, and the food it's made is sucrose. The sucrose moves towards the vascular bundle into the companion cell. It then goes into the sieve tube cell, and the sieve tube cell then moves it all the way down in the phloem to the root. When it gets to the root, the sugar or sucrose goes out through the companion cell and it goes into the root cells. And the root cells either use it up for energy or they store it for springtime, and that food would then be used for growth to start in spring. Now, the reason why phloem has companion cells is substances don't always move down. Sometimes, if a flower or a fruit is being made, the leaves make food and they send it up to the fruit in the phloem. And that's why you have companion cells, so that if necessary, the sugar can be moved up into the fruits. Before your brains burst too much, let's take a bit of a break, and when we come back, we will look at how to answer questions on these particular aspects of plant structure and functioning. See you later. Welcome back from your break, and we are now going to be looking at questions that can be asked on all aspects of plant structure and functioning. Questions often start off simply where you are given a picture. In this case, it's an example of a pitometer, and you're asked, what does this piece of apparatus actually do? That's one of the straightforward ones, and of course, that tests the rate of transpiration. Then questions get a little bit more tricky, because if we look at this example, it shows us drawings of the surface of two leaves, and the dark things are the guard cells. And if you have a look, leaf A and B have a different distribution. Leaf B has got a lot more stomata, and you need to be able to say, okay, if it's got more stomata, it means it's going to be losing more water, all sorts of things like that. And questions will be asked to check to see if, have you picked that up? So, if we have a look, it starts off with easiest questions. Okay, what is W? W is a pore, so it could be either C or D, and it's clearly the pore because it's pointing to the hole. So the next ones we need to look at, okay, what is X? X is a guard cell, so that is correct. So your correct answer there is C. Then, the next question now, this is testing your understanding, in what environment would this plant, leaf A, survive in? Okay, and if we look, it's got fewer stomata, so that tells us that it is adapted to a drier environment. If there are fewer stomata, it also means that less carbon dioxide can get in, so it would survive in an environment that has got a lot of carbon dioxide. So we can say, okay, it is drier, so that would work. Wetter, nope. Drier, yes. Wetter, nope. Then, would it survive if there was less carbon dioxide? Because it's already got less stomata. So if there was less carbon dioxide, it wouldn't be able to get in enough carbon dioxide. If it's got more carbon dioxide, it would be able to survive. So the correct answer there is C. Then, sometimes you're given photographs taken under a microscope and you're asked to identify each different type of plant organ. So, let's have a look at this. It's a plant part taken under a microscope and answer the questions. Is this a cross or a longitudinal section? And you should know cross-section is cut across like this. Longitudinal section is lengthways. And the only way you would get something like this is via a cross-section. Then, is it a root or a stem? And the key to that would be, look for the xylem. If the xylem is in separate vascular bundles, it's a stem. If the xylem is in the center, then it would be a root. And we can see clearly that there are separate vascular bundles, so that is obviously a stem. Then identify the layers. Okay, so let's have a look. Layer number 2, that's that. That's our bundle cap. Layer number 3, which is your phloem and cambium. And layer number 4, which would be the xylem. What is the function of layer 1? That's just underneath the epidermis, and that's where the parenchyma is. So its function would be support. Next one is a drawn picture of a leaf with a whole lot of the different parts labeled. Let's have a look. Okay, give the letter and name of the tissue responsible for transporting water. Okay, so you now need to look, and transporting water, that is T, the xylem. Transporting manufactured food away, that is Q, the phloem. And then the reason why it's 4 marks is because there's a mark for the letter and a mark for the name. Then, next thing you have to give the letter and name of the main photosynthetic tissue. And the main photosynthetic tissue here is R, which is the palisade mesophyll. Then it takes you further because you now have to describe 2 ways in which these cells are adapted for maximum photosynthesis. And that's for 4 marks. So you would have to talk about the fact that they are elongated, closely packed, few air spaces, filled with chloroplasts, directly underneath the upper epidermis. Any 4 adaptations which make them more effective at their job, which is maximum photosynthesis. Then, still on the diagram of the leaf, explain 2 functions of the cells labeled P. Okay, so P, that is the epidermal cells, so that would be protection. If they have a cuticle, it would be lessening of water loss. It could also be they're transparent to allow light in. Any 2 functions. Then, 4.5, name the cells labeled W and V. Okay, so V, that is those cells there, and W, that is your ordinary epidermal cells. And then you have to tabulate 2 structural differences. Okay, in other words, you need to say how are they different in structure. And obviously, if we look at the size, the guard cells, they're little cells. What is their shape like? They are bean-shaped. These cells are very often rectangular. what's different about these walls of the stomata? They've got thick inner walls and thin outer walls. These have got thicker outer walls and thin inner walls. Okay, so it's basically any two structural differences. The next one we're looking at is label S, and label S is pointing to the spongy mesophyll, and you have to give the primary function of this layer of cells, and because of the fact that there are so many air spaces, it's obvious that the function of this layer of cells is basically for gaseous exchange. Now, you will have noticed that as we've been going through the questions, gradually a little bit more has been incorporated, because we started off with straightforward, what is this? Now we're bringing in all sorts of things, and this next question we're looking at is an example where we are also including different experiments that can be done, as well as the root, stem and leaf and the water movement through the leaf. Now, a question like this, to be able to do it properly, you really need to be able to understand and put everything together. So, let's have a look at this. Okay, we have a picture, and this is obviously, this is below the soil. On this side of the diagram, we have a picture of a plant with a root, the bottom here, below the soil surface. It shows root hairs, water moving into root hairs, moving up through the xylem. And B is obviously pointing to the stem because the water is moving up the xylem and it's going into the leaf. When the water gets into the small xylem vessels in the leaf, it leaves the xylem. Some goes into the cells, some goes out through the process of transpiration. Then, other pictures show different organs found at different parts of the plant and an experiment. And the questions will need you to link everything together. So, always starts off with easy questions. Identify A, B, C, and D. A is the root hair, B is the xylem going all the way up, C is an epidermal cell, and D is your stoma. Then, describe the way in which part A is adapted. A is your root hair, so here again you are talking about an increase of surface area. Then, 3 adaptations of tissue B. And here's tissue B, and tissue B is your xylem. So you need to discuss 3 ways xylem is adapted, for example, no cross walls, thick walls to prevent collapse, no cell contents joined end to end, things like that. Any 3, because it's out of 3 marks. Then, describe the function of part D, and part D is the stoma, and you would simply say opens and closes to be able to let water out of the leaf or to let gases into the leaf. Okay, then it starts getting a bit more complicated because you now have to draw up a table comparing tissues E and F. The next question asks you to compare tissue E and F, and if we look at the picture here, tissue E is your palisade mesophyll and tissue F is your spongy mesophyll, and you have to compare them in terms of shape, round or rectangular, air spaces, present or absent, number of chloroplasts, lots or a little, and the main function, which is gaseous exchange or photosynthesis. Next question asks us to look at the process that occurs in part A, and here's part A, which is basically your root hair. Which process can be linked to the apparatus here? And the apparatus here is a diagram on osmosis. So answer to 5.6 is osmosis. Then if we look at 5.7, which part of the diagram 4, which is the bottom one here, corresponds to the following diagram 3, and your dialysis sheet corresponds to the cell membrane because they are both semi-permeable, and the sugar solution links to the vacuole, the water and the sugar in the vacuole. The next question looks at how much water different plants lose under different conditions. Okay, and there's a whole big explanation of an experiment in which a plant is placed in a test tube of water and it's placed in the sun for 5 hours. Then the same plant is put inside a plastic bag and placed also in the sun for 5 hours, but the plastic bag changes one environmental factor, which is humidity. Then the next question, 6.2, why is it better to use the same plant? And we've come across that before because different plants would have a different number of stomata. The next question looks at the graph of the results, and it shows that A loses a lot more water than in situation B when it's placed in a plastic bag. And what you have to do is explain how plant B loses a little bit of mass because it loses some water, but then the plastic bag stops it from losing any more water because the concentration or the humidity outside in the plastic bag would be very high. Then 6.4, what other conditions might affect the outcome and thus need to be controlled? And those are all the other factors that affect osmosis, which is temperature, light intensity, and wind. Then the last question, how would a farmer grow leafy vegetables, conserve water? And that would obviously be, he would put his plants in a greenhouse because that would keep the water that's being transpired, it would be kept in the greenhouse, it would increase the humidity, so they lose less water. I hope that this revision lesson has helped you a little bit and you will feel more confident when you actually face questions that at first look quite difficult, but if you look at them step by step, you can get there. I hope this series of lessons has been of value to you. See you next time.